I'm Naima Pyarali, and I'm a senior at Albuquerque Academy. Today, I'm going to be talking about the research I did with Lumiere. But first, I just want to thank my mentor, Ariana Broad, who is a PhD student at Cornell. I couldn't have done it without her. Now, I did a biochemistry literature review, and the question was, what DNA repair response is elicited by transposable elements, specifically CRISPR-Cas12a? Now, let's move on to define some key terms. Next slide, please. So DNA damage happens daily due to UV exposure, carcinogens, and more. The body combats this damage with a DNA damage response, DDR. There are two types of DDR. One type, such as non-homologous end joining, or NHEJ, depicted on the left of the screen, is very error-prone and can result in some code deletion, but it's the quickest response. The second DDR, homology-directed repair, HDR, depicted on the right of the screen, is less error-prone but requires the use of more proteins and takes longer. Transposable elements, or TEs, are mobile genetic elements that can integrate into the host DNA. There are TEs native to the host genome, which are helpful for creating genetic diversity. However, retroviruses such as HIV also use TEs to integrate into the code in order to be less treatable. TE integration elicits a DDR, which I'll be exploring. But what does CRISPR have to do with it? Well, the most popular and used CRISPR system, CRISPR-Cas9, can minimally modify genetic code. However, there are many mutagenic and off-target results where it modifies code it's not supposed to. The hope is to explore a new system, CRISPR-Cas12a, which is a transposable element that can significantly modify DNA in hopes of having a low mutagenic rate from DDR. By studying other TEs, we can better hypothesize what Cas12a will do. So let's see what my research showed. Next slide, please. So through a literature review of over 20 papers on each type of TE, I found that 1, retroviruses depicted on the left elicit a stable HDR response, which leads to low mutagenic rates and easy integration. In the middle, these native TEs prefer error-prone NHEJ, which leads to mutations that create the genetic diversity these TEs aim for. However, if there's no DNA PK and Q70, proteins that are needed for NHEJ to occur, then the less error-prone HDR can occur during integration. Now, the third set on the right, the CRISPR-Cas9, prefers the error-prone NHEJ because it uses the DDR at the target DNA to create a mutation in order to change the code slightly. However, this does not always work, and there are many off-target results, as I said. Additionally, when Cas9 contained exogenous or external DNA and proteins, it elicited a stable HDR response, where you can see on the most right of the Cas9 depiction. CRISPR-Cas12a is a very similar system to TN7, a TE found in E. coli. In prokaryotic research, it seems CRISPR-Cas12a does not elicit a DDR. But what does that mean? But that does not mean that will have the same results in humans. So what did I conclude with this research? Next slide, please. CRISPR-Cas12a may react 3 different ways in humans. 1, there will be no DDR as seen in E. coli, which is highly unlikely due to the other TE research. 2, the DDR will be like the native TEs. This means they will prefer NHEJ, the highly mutagenic response, which can be troublesome as this will lower the efficacy of CRISPR-Cas12a. However, if this is the case, there is hope that certain conditions can be controlled where HDR can be elicited, as seen with the previous research I talked about. And 3, it will act the same way as retroviruses in Cas9 when there's an exogenous or external DNA and elicit a less error-prone HDR response. This is the most likely possibility since the Cas12a system will contain foreign DNA, which the body usually reacts to with a more reliable HDR response. With this conclusion, there's hope that the CRISPR-Cas12a system will be successful in genetically modifying human DNA with a high accuracy. Thank you.